Good morning, everyone, and welcome. I'm delighted to have you here today. My name is Dr. S.K. Singh, and I hold a PhD in this field. Today, I will be presenting on cleanrooms designed for pharmaceutical facilities as per cGMP standards. In this session, we'll explore the key principles of designing cleanrooms that meet regulatory requirements, ensuring safety and quality in pharmaceutical manufacturing. I look forward to sharing valuable insights with you. Let's get started. So what exactly is a cleanroom? Well, it can be understood through two main areas, control and design. The control aspect refers to the space where we measure and regulate the number of airborne particles, ensuring the environment remains as clean as needed. It also involves managing other environmental factors like humidity, temperature, and pressure to maintain optimal conditions. On the other hand, the design aspect focuses on how the cleanroom is constructed. It's built as an enclosed space, carefully designed to minimize the introduction, generation, and retention of particles. This combination of proper control and thoughtful design is what makes cleanrooms effective. Now, why do we need these cleanrooms? They're essential for controlling airborne contamination to levels that make it safe to perform activities that are highly sensitive to contamination. You'll find cleanrooms in industries like aerospace, microelectronics, pharmaceuticals, medical device manufacturing, healthcare facilities such as hospitals, and even in food production. All of these fields require strict control over their environment to ensure safety, quality, and precision. So, understanding what a cleanroom is and how it functions sets the foundation for appreciating their critical role across various industries. Current Good Manufacturing Practices, or cGMP, are a set of regulations that are enforced by authorities like the U.S. Food and Drug Administration. These regulations are in place to make sure that products, especially pharmaceuticals, food, and medical devices, are consistently produced and controlled to meet high-quality standards. The current in cGMP is really important because it emphasizes that companies need to stay up to date with the latest technologies and systems. It's not just about following old rules, but making sure that practices evolve with advancements. These practices cover all aspects of production, everything from raw materials and equipment to facility conditions, staff training, hygiene, and record keeping. Every step matters. One of the key goals of cGMP is to prevent contamination, mix-ups, and errors. How? By requiring proper documentation, validated processes, and regular quality checks at each stage of manufacturing. Following these practices is essential because it ensures that products are safe, effective, and of consistent quality. Ultimately, cGMP helps protect consumers and make sure that what's on the label is what they're actually getting. So, in the bigger picture, cGMP is a cornerstone of quality assurance, regulatory compliance, and public health. It's about maintaining trust in the products that millions of people rely on every day. Let's take a moment to understand what a particle is in the context of cleanrooms. A particle is essentially a solid or liquid object that, for classification purposes, falls within a specific size range, between 0.1 and 5 micrometers. These particles are important because their presence can impact the cleanliness level of the environment, especially in controlled spaces like cleanrooms. Now, moving on to occupancy states, which describe the different conditions of a cleanroom depending on its operational status. First, we have the as-built state. This is when the installation is complete, all services are functioning properly, but there are no production equipment, materials, or personnel inside. It's essentially the room in its ready state, waiting for the next phase. Next is the at-rest state. In this phase, the equipment has been installed and is operating as agreed upon by the customer and supplier, but there are no personnel present. Think of it as the room being prepared and ready for operation, but nothing is actively happening inside at that moment. Finally, we have the operational state. This is when the cleanroom is fully functioning according to its intended purpose. The equipment is running, the personnel are present and working, and everything is in compliance with the required standards. Understanding these definitions helps us appreciate how cleanrooms are monitored and maintained at different stages. ensuring they meet the strict cleanliness standards necessary for their specific applications. Let's take a closer look at the classification system. The ISO classification number, which we refer to as N, ranges from 1 to 9, with 1 being the cleanest and 9 the least clean. This number helps us categorize the cleanliness level of the air in controlled environments. Now, the particulate cleanliness of the air isn't just a single measure. It depends on different occupancy states. We define cleanliness in one or more of 3 states. As built, which is the environment right after construction. At rest, meaning the space is not in active use. And operational, when the space is actively in use with processes running. For each of these states, there is a maximum allowed concentration of particles, which we denote as CN, and this value depends on the particle size, D, that we're considering. The classification essentially sets the upper limit on how many particles of a given size are permitted per cubic meter of air. So, when we talk about classification, we're referring to the level of airborne particulate cleanliness. A way to quantify how clean the air is by specifying these maximum particle concentrations. There are 9 ISO classes, ranging from ISO 1 through ISO 9. Remember, ISO 1 is the cleanest environment, with the strictest particle limits, while ISO 9 allows for the highest concentration of particles and is considered the dirtiest. This classification system is fundamental because it guides the design, validation, and operation of cleanrooms and controlled environments across various industries. It ensures that we maintain the appropriate cleanliness levels for specific processes and products. Let's take a closer look at the ISO classification for cleanrooms. As you can see, each ISO class specifies the maximum number of particles of 0.5 micrometers or larger per cubic meter of air. For example, ISO Class 1 allows only up to 10 particles per cubic meter, making it suitable for the most critical environments. The table also shows the recommended air changes per hour, which vary depending on the class. For ISO 1 and 2, you need between 360 and 600 air changes per hour to maintain the cleanliness standards. Ceiling coverage is another important factor, indicating how much of the ceiling area should be covered with HEPA or ULPA filters to ensure proper air filtration. For ISO 1, nearly 100% coverage is required, whereas for lower classes, the coverage can be less. Overall, these parameters, particle count, air changes, and ceiling coverage, are key to designing cleanrooms that meet specific environmental standards. They help us control contamination levels effectively, depending on the class of cleanliness we need to achieve. Let's take a closer look at the PIC-S guidelines for cleanroom classification. On this slide, we see a table that outlines the maximum permitted number of particles per cubic meter, depending on the cleanroom grade and whether the equipment is at rest or in operation. Starting with grade A, which is the highest level of cleanliness, the limits are quite strict. At rest, the maximum allowed is 3,520 particles that are 0.5 micrometers or larger, and just 20 particles of 5 micrometers or larger per cubic meter. When the equipment is in operation, these limits remain the same for the smaller particles, but the larger particles are also limited to 20 per cubic meter. Moving to grade B, the particle limits are a bit more lenient. At rest, the maximum is again 3,520 particles of 0.5 micrometers or larger, but the limit for 5 micrometers increases to 29 particles. In operation, the smaller particles stay at 352,000, but interestingly, the larger particles are still limited to 29 per cubic meter. For grade C, the limits are much higher, reflecting a lower level of cleanliness. At rest, you can have up to 352,000 particles of 0.5 micrometers or larger, and 2,900 of 5 micrometers or larger. In operation, these numbers increase significantly, to 3.52 million for the smaller particles and 2,900 for the larger ones. Finally, grade D has the highest permissible particle counts. At rest, up to 3.52 million particles of 0.5 micrometers or larger, and 29,000 of 5 micrometers. When in operation, the limit for 0.5 micrometer particles remains at 3.52 million, but for larger particles, the limit is set at 29,000, though some parameters are not defined explicitly. The key point here is that these classifications help ensure the controlled environments necessary for different pharmaceutical or biotech processes, with stricter limits for higher-grade cleanrooms. This table highlights how particle counts are managed and monitored to maintain the required level of cleanliness throughout different operational states. Let's take a closer look at this table, which outlines the classification of cleanrooms according to cGMP guidelines. As you can see, the table is divided into different grades, from A to D, each with specific air quality and microbial limits. Starting with grade A, which is the highest level of cleanliness, the maximum particles of 0.5 micrometers at rest and in operation are both limited to 3,520 particles per cubic meter. The microbial limit is less than 1 CFU per cubic meter of air, and settle plates, contact plates, and glove prints all show less than 1 CFU. This level is critical for environments requiring the utmost sterility, such as aseptic manufacturing. Moving to grade B, the particle limit remains the same at rest, but in operation, it increases significantly to 352,000 particles. The microbial limit is slightly higher at 10 CFU per cubic meter. Settle plates and contact plates have limits of 5 CFU, and glove prints are also limited to 5 CFU. This grade supports environments where aseptic work is ongoing, but slightly less critical than grade A. For grade C, the particle count at rest jumps to 352,000, and in operation, it rises further to 3.52 million particles. The microbial limit here is 100 CFU per cubic meter, with settle plates at 50 CFU, contact plates at 25 CFU, and no data for glove prints. This grade is suitable for less critical operations, but still maintains a controlled environment. Finally, grade D allows even higher particle counts, up to 3.52 million at rest, with no defined limit during operation. The microbial limit is 200 CFU per cubic meter, and settle and contact plates have limits of 150 CFU respectively. Again, no data is given for glove prints. The key takeaway here is that as we move from grade A to D, the allowable particles and microbial limits increase, reflecting a decrease in environmental control. These classifications help us ensure that each area meets the specific requirements of the processes taking place within them, aligning with cGMP standards to maintain product quality and patient safety. Let's talk about the guidelines for the required number of air changes in different types of rooms. For Class A rooms, the recommended air changes per hour are between 240 and 480. This high range helps ensure a very controlled environment, often needed in highly sensitive settings. Moving to Class B rooms, the guideline drops to about 60 to 90 changes per hour. These rooms still require good ventilation, but not at the same intensity as Class A, and for Class C rooms, the recommended air changes are even lower, around 20 to 40 per hour. It's important to remember that these numbers are just guidelines, not strict regulations. They can vary depending on different sources and specific circumstances. The key concept here is that higher class rooms, which typically require more stringent control over air quality, need more frequent air exchanges. This helps maintain a safe and clean environment. Ultimately, understanding these guidelines helps us design ventilation systems that support health, safety, and overall comfort in various settings. Let's take a closer look at Slide 10. Here, we're discussing two important international instruments, the Pharmaceutical Inspection Convention and the Pharmaceutical Inspection Cooperation Scheme, or PIC-S. These are essentially agreements between countries and their pharmaceutical inspection authorities to promote harmonization and collaboration. Focusing on PIC-S specifically, it acts as a tool to enhance cooperation among regulatory bodies and the pharmaceutical industry, particularly in the area of good manufacturing practices, or GMP. The goal is to ensure that manufacturing processes meet consistent quality standards worldwide. Now, moving into the classification of clean areas, we start with Grade A. Think of this as the highest risk zone. It's where the most critical operations happen, like filling aseptic products, working with open ampules and vials, or making aseptic connections. These areas are typically provided by a laminar airflow workstation, which maintains a very controlled environment with a homogeneous airspeed, generally between 0.36 and 0.54 meters per second. It's important to note that in some cases, such as in closed isolators or glove boxes, we might use unidirectional airflow with lower velocities to maintain sterility. This helps minimize contamination during these sensitive procedures. Next, we have Grade B, which serves as the background environment for Grade A. Think of it as the environment that supports aseptic preparation and filling. It's slightly less critical, but still requires a clean, controlled setting. Finally, Grades C and D are even less critical. They are clean areas designated for manufacturing less sensitive stages of sterile products. These classifications help ensure that each part of the process occurs in an environment appropriate to its risk level, which ultimately upholds product quality and safety. So overall, these classifications, guided by PIC-S standards, help us maintain a structured, consistent approach to environmental control in sterile manufacturing. Let's talk about the clean areas. These are designated spaces where strict control of contamination is essential. Entry into these areas is only through airlocks, which serve as a buffer zone for personnel, equipment, and materials. This helps prevent any unwanted particles or contaminants from entering the clean zone. Additionally, the air supplied to these areas has to pass through filters with the appropriate efficiency to ensure the air remains as clean as possible. Within these clean areas, different operations are carried out in separate zones. For example, component preparation happens in one part, product preparation in another, and filling operations are conducted separately as well. This separation helps maintain the integrity and cleanliness of each process stage. When we look at manufacturing operations, there are two key points. First, the product may undergo terminal sterilization, which is the final step to ensure sterility. Second, some or all stages of the process are conducted aseptically, meaning under sterile conditions, to prevent contamination throughout production. To meet the necessary in-operation conditions, these areas should be carefully designed so that, even when in use, they can achieve certain air cleanliness levels. This is especially important in the at-rest state, when the area is not actively in operation, but still needs to maintain high cleanliness standards. Finally, the classification system we refer to here is PIC-S, which helps define and standardize these cleanliness levels. Overall, these measures are vital to ensure product safety and quality in pharmaceutical manufacturing. Let's now focus on the classification of terminally sterilized products under PIC-S guidelines. Typically, the preparation of components and most products should be carried out in at least a Grade D environment. This ensures a basic level of cleanliness and control. However, when the product is at a higher or unusual risk of microbial contamination, we escalate the environment to a Grade C, providing a more stringent level of control. Filling of products that will undergo terminal sterilization is generally done in a Grade C environment, especially if there's an increased risk of contamination. But in cases where the product is at an unusual risk, such as with sensitive or high-risk products, filling is performed in a Grade A environment with a Grade C background. This means the actual filling happens in a very clean, controlled environment, with surrounding areas maintained at a slightly lower grade to support the process. When it comes to preparing and filling ointments, creams, suspensions, and emulsions that will undergo terminal sterilization, these should also be prepared in a Grade C environment before the sterilization process. This step ensures that the product is handled under controlled conditions to minimize contamination risks. Moving on to aseptic preparation, components after washing should be handled in a Grade D environment, which is suitable for initial cleaning. For handling sterile starting materials that are not sterilized or filtered beforehand, a Grade A environment with a Grade B background is required. This provides the highest level of cleanliness necessary for aseptic handling. If sterilization or filtration has already been performed on the starting materials, handling can be done in a Grade C environment. When it comes to handling and filling aseptically prepared products, a Grade A environment is essential to maintain sterility and prevent contamination. Finally, the transfer of partially closed containers, such as those used in freeze drying, should be done either in a Grade A environment with a Grade B background or within sealed transfer trays in a Grade B environment. This ensures the integrity of the sterile process is maintained throughout transfer. By understanding these classifications and their appropriate environments, we can ensure our manufacturing processes meet the required standards for safety and efficacy. Let's wrap up with a quick summary of what we've discussed about clean rooms. These environments are designed to maintain very strict control over air quality, temperature, humidity, and pressure to keep everything as sterile as possible. To achieve this, high-efficiency particulate air, or HEPA filters, are used to remove particles from the incoming air, and airflow systems, often laminar flow, ensure a continuous supply of clean, uncontaminated air. It's also crucial that personnel entering these clean rooms follow strict gowning and hygiene procedures. This helps reduce the risk of contamination and keeps the environment safe for product manufacturing. In pharmaceutical settings, clean rooms are classified based on the level of cleanliness required, according to cGMP standards. We have grades A, B, C, and D. Grade A is reserved for critical operations like aseptic filling, where the highest level of cleanliness is necessary. Grade B provides the background environment for aseptic areas, while grades C and D are used for less critical steps, such as preparation and washing. To ensure ongoing compliance, regular environmental monitoring is essential. This includes checking air quality, surface cleanliness, and personnel hygiene to make sure particle and microorganism levels stay within acceptable limits. Ultimately, clean rooms are vital in pharmaceutical manufacturing because they safeguard product safety, quality, and sterility. This protects patients from contamination-related risks and ensures that the medicines they rely on are safe and effective. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional information you might need. Feel free to contact us at any time. And with that, I want to sincerely thank you for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, we're always here to support your success. Thank you once again.